Okay, so we covered the reactions of amides and that sort of thing. What I want to do now is talk about some of the most important amides, and those are peptides. Okay, so peptides are, it's kind of a weird word. So peptide is sort of another word for amide bond. It's one of these things where amides were discovered at the same time that peptides were discovered, but they weren't discovered by the same people. So proteins, it turns out, are polypeptides. Polymers of amide bonds. So the amide bonds are what kind of connect the monomers together to make the polymer chain. Now, that's interesting. Why wouldn't they just call them polyamides? Polyamides also exist, and that's another useful term, but this usually, polyamides usually describe industrial polymers usually describe industrial amide polymers. Okay, so let's get back to polypeptides. So polypeptides, which again, the term is protein, use alpha amino acids as monomers. Use alpha amino acids as monomers. And so let's go ahead and remind ourselves what alpha amino acids are. So we have this alpha position that has an amine and a carboxylic acid. So we have our amino group, and we have our acid group. What I want to do is just insert one page here. Perfect. Okay, we have our amino group and our acid group, and we have this R group. Now the R group this varies depending on the amino acid that we're looking at. So the R group varies depending on the amino acid that we're looking at. Let's see, I actually don't want that page. So I want to, sorry, I'm just going to delete this page really quick. So, well, okay, I'll delete it later. So the R group varies depending on the amino acid that we're looking at. Now what I want to do is transition, and I wanted to include this in the notes so that you all could print it out. I just grabbed a picture on the internet of the common 20 amino acids that are typically used in biology. And so we have the names, and then we have, so for each has a name, like we have alanine, glycine, that sort of thing, all the way through. And we also have the one-letter code. So the one-letter code sort of is different among all of these amino acids because we have to put them together in a sequence, a polymer sequence, to make a polypeptide or to make a protein. And as many of you know that have taken some biology, the sequence of those amino acids are critical for determining the properties of the protein. Now, in addition to the one-letter code, we've got a three-letter code, which could be ALA, GLY, ILE, depending on the amino acid. We just have so many different ways of kind of thinking about and communicating the structure of these amino acids. Okay, so in each of the cases, the R group is sort of different. So let's remind ourselves about the amino acid structure. I'm considering the neutral form for all of these. We'll get to the zwitterion here in a second. But in alanine, for example, alanine, the R group is equal to a CH3. So we just have a methyl group hanging off of the chain. In, let's pick valine, R is equal to an isopropyl group. And we get this one now. We could have a benzene ring as our R group. Now it's a benzene ring with a CH2 attached to it, so there's our R group, and that would give phenylalanine. And then my favorite one of all is serine, which has a CH2 to an OH, so there's serine, and you can see the structure for serine right here. There's an awkward one that actually, that's also one of, I'm a big fan of a lot of these amino acids, I'll be honest with you. Some of my favorite molecules. But we could also have this structure, and this structure is in the top row. This is proline. Come on. Proline. So proline is one of the most interesting amino acids. It's actually very useful as a catalyst for a lot of organic transformations. So they call it an organocatalyst, which is kind of fun. Okay, so there are some structures of some amino acids. And now what we can do is we can stitch these together to make polymers of a polyamide that are derived from these amino acids. So polypeptides have the general repeat unit where we've got a carbonyl at one end, and then we've got our nitrogen at the other end. The nitrogen has one H, and then we have our side chain, and let's see, we've had to rotate some things around, so I would wager that we go like this with our side chain into a dash. Okay, so, and we'll get to that stereocenter in a moment, but this is our repeat unit for a polypeptide. So we have our alpha position, we have our amine, our carboxylate, and then our side chain that's hanging off of the molecule that defines the identity of the amino acids. Okay, so we can connect these together by way of a series of reactions. So let's go ahead and take alanine. So here's alanine. And we're going to react this with glycine. And we can react this with phenylalanine. Okay, and we can make a polypeptide. Now, how do we polymerize these things? That's usually done with enzymatic machinery within the ribosome that allows for, this is kind of trans protein expression, I'll just say. I was about to say, is it translation? Yeah, it's probably translation, converting mRNA into proteins. But we do a polymerization with enzymes. This isn't a biology class, so I'm not going to focus on the enzymatic machinery more than just kind of telling you what it is. So let's think ribosome is what I believe we want to see that happen, where that's going to happen. Now the enzyme is going to convert the structure into a polymer where we have our alanine first, and that's going to make an amide with the glycine, which is going to make an amide with the phenylalanine. So here's our phenylalanine. We can tell because we've got that CH2 into a benzene ring. When we have two hydrogen atoms, that then becomes glycine. And then over at the end here, we've got our valine. Now with the peptide chain, we typically have an end to the polymer. This is the end with the free NH2. That is our N terminus. And that's in contrast to our carboxyl end of our polymer. That is the C terminus. Now for this polymerization to occur, we're going to lose as many H2Os as we have amino acids minus 1, where N is equal to number of amino acids minus 1. So if we have 3 amino acids to build up our polymer, we're going to lose 2 water molecules, and each of those water molecules results from formation of an amide. Okay, so it's important to note, though, that this reaction, the formation of this polymer, is a complicated reaction that again involves enzymatic machinery. Enzymes is our favorite catalyst, and that's because amino acids typically exist as zwitterions. So these things exist as zwitterions. This helps with water solubility. By having these charges present, we can now more effectively hydrogen bond with water and get our stuff solvated, get our amino acids solvated into the solvent of life, which is water. But the CO- and NH3 plus make for bad reaction partners. In amide bond forming events. So we have to kind of, it turns out we have to attach the amino acid to tRNA, translational RNA. So what this looks like then is we have some mRNA, and that interacts with tRNA by way of the ribosome, and translation occurs to make proteins. So we have AAs, just the amino acids are literally hooked onto the tRNAs, and then they're sort of strung together where we have amino acids forming a string together, which makes a protein. Okay, so proteins are, are an interesting polymer because proteins are a sequence-specific polymer, and really that's true of DNA and RNA. So these are sequence-specific polymers. Now, it's important to appreciate that this is very weird. It's very weird to be a sequence-specific polymer because when we do polymerizations in the lab, or when we've talked about polymerizations before, like making polyvinyl chloride and that sort of thing, we use the same monomer over and over again. We just react it, blah, 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 blah. You have a bunch of chlorines at the end, you have polyvinyl chloride. You can replace these side chains with something, but it's not some things, okay? It's all the same. Here, we have roughly about 20 amino acids that are present and used in various kind of orders and arrangements to give us a multitude of possible polymers that could be formed, right? Like nearly, a really big number, probably an infinite number of possible sequences. Obviously, if you don't put a cap on it, it is an infinite number. But anyway, a big number of possible sequences, which could result in a big number of possible polymers, which thankfully enough, because there's a big number of possible polymers, there's a big number of possible properties that can result from these polymers, giving rise to the diverse function of proteins, as opposed to DNA, which is mostly just kind of the informational storage system. Proteins can do everything from allowing muscles to move, to moving I mean, they transform so many different, excuse me, metabolites in our body. Yeah, they're just so versatile. They're my favorite biomolecule, but all biomolecules are the same. We don't have to worry too much about that. I think it's worth mentioning just ever so briefly on the classic approach that chemists used to make proteins. And so let's just mention that briefly. So chemists used to make proteins. Well, they still do. They still do. Let's just, you know what? I'm going to gently cross that out. It is an aging technology. I realize the grammar's weird there with the 2 in there, but chemists used to make proteins using solid phase peptide synthesis. And they still can. There's a lot of people that do. There's an important alum that does make peptides this way. And they do so because they're using unnatural amino acids. So what they do is they use solid phase peptide synthesis. This takes advantage of what's called a catch and release purification protocol. Okay, so let's talk about it briefly. So what we're going to do is we're going to take our amino acid. Let's go ahead and just draw it as the zwitteran so that we can be sort of thinking chemically about this. And I'm going to append this to some sort of group that's attached to solid resin. So step 1 is add the amino acid to a solid resin. Now, solid resin is interesting because it's insoluble. So this resin is literally something that we can see. It actually makes like a bed and that sort of thing. But we attach this to we attach the amino acid to the resin. And what we get out is sort of a resiny ester. And I guess R prime on the side chain, sure. Anyway, the R over here is supposed to stand for resin. The resin is usually attached to the molecule by way of like a linker by a long chain linking unit, which I'm trying to capture here. Okay, when we do this, the NH3 now becomes NH2, and this is reactive. Okay, so what we're going to do is we're going to add amino acid number 2. And we're going to have amino acid number 2 protected at nitrogen. So I'm going to put PG here for protecting group. Okay, now the protecting group can be what's called a Bach group or an FMOT group. Just mentioning these in case you want to refer to these notes later on. We have had many alum kind of practice this technique, and it's a lot of this stuff is still useful. Okay, so anyway, this is protective, and what that means is that this nitrogen is unreactive. And so what we can do is we can expose these two to an amide bond forming reaction. And that gets us into step 2. I'm going to zoom out. I just want to make sure I'm still kind of formatting things. So step 2, make an amide with a protected amine, amino acid. So when we make the amide, what we're doing is we're taking the reactive nitrogen and we're engaging that with the carboxylic acid on the unreactive nitrogen-containing amino acid. So the second amino acid gets connected because the carboxylic acid is present, but the amine is protected. By having the amine protected, we avoid some unwanted side reactions where we scramble which nitrogen adds to the carboxylic acid. So we're going to do like a carbodiamide type of reaction that we talked about with amide chemistry. And we're going to get an amide bond here. So we have a new bond by connecting the nitrogen to the amide. Now what we've done is we make the amide. Sorry, let me get a new page here. And then in step 3, we deprotect and wash away impurities. And when we deprotect and wash away impurities, we get to a resin that looks different than our starting resin. In particular, the nitrogen is now adducted to a carboxyl group. And that nitrogen was red, and I'm just kind of painting everything a highlighted color so we can keep track of everything. And we add another amino acid. So we unprotect the amine, and we wash away all of the impurities. And the reason why we can wash away all the impurities is the growing the chain that's on the resin. That resin's insoluble. So if we rinse everything with some sort of cleaning solvent, our resin and the stuff attached to it stays in solution, but all of the impurities wash away. Okay, now this unprotected group is ready for the next amide bond reaction to form. So this is ready for the next amide bond reaction to form, and so you could repeat over and over again to make a long, and I'm going to put ish, peptide chain. Okay, so what we're doing here is we're doing a sequence of amide bond forming reactions where we make an amide bond forming reaction using a carboxylic acid with an amine, but that amine's protected, so that amine doesn't get in the way. So we use the carboxylic acid with the unprotected amine. We mix those together by making an amide bond reaction, and all is good. Well, to make this sustainable to do, or like efficient to do on a big scale to make big proteins, we have to be able to remove the impurities effectively, because if we had to do a really hard purification at each stage, nobody would pick up this technique. So to do that, to kind of make the purification at all practical, we do what's called catch and release, where we build a long polymer chain on this resin. The resin can continuously be rinsed. And then after we make our longest polymer chain, big arrow, we remove protein, or our poly amino acid polypeptide, from the resin. And then we get empty resin that can often be recycled. So that's an efficiency. And then we have amino acids strung together, and we have a protein. Now the protein, so this was great because what we didn't have to use any like tRNAs, we didn't have to use any biomolecules. We just grabbed some protected amino acids, and there's only 20 of them. So we just buy some protected amino acids of all flavors, and we put them in the particular order that we want. And we can then grow the polymer with a specific sequence, which again is really hard, using common laboratory techniques without a super intensive purification protocol. The problem is we have a limits to maybe 20 amino acids. So after about 20 amino acids, we can't really add anything else. So how do we make proteins that we want? Well, we've been actually doing this for a while now. I mean, the revolution has been tremendous in the past 20, 25 years, where what we do is we just rely on biology. Biology is great at making proteins. It just takes a string of DNA, transcribes it into RNA, which then uses RNA, then goes into the ribosome and uses the amino acids that are bound to tRNAs to synthesize a sequence of amino acids. And it does so very efficiently, just using biological stuff that's already lying around. This has been happening, since biology really started taking off in terms of evolution. And the proteins can be enormous, like hundreds to almost, to over 1,000 amino acids in length, giving rise to a huge diversity of proteins that can be formed. So what we needed to do was just get good enough with DNA to use that technology to build the proteins that we want. And that's exactly what we do. We just install a gene of interest, a sequence of DNA into a cell, tell the cell, hey, focus on making this. I know you want to survive and do all that stuff, but you're just a bacteria cell. I'm not too worried about you in the grand scheme of things. Thanks for helping me out this time, though. And it's going to use all of its protein synthesizing machinery to generate our protein of interest, which we can then use for numerous things, like understanding biological pathways more and more, to even developing the next generation of catalysts for complex organic reactions that would otherwise not be practical. And whenever you can use enzymes to do organic chemistry, all of a sudden, everything gets a lot greener. That is in terms of the how well behaved this plays with the planet Earth, because we can use water as a solvent. I mean, we have to use water as a solvent if we use proteins. And the proteins are some of the most efficient catalysts. We can use such small amounts of them to accomplish these transformations. And they're going to do so with exquisite selectivity. Oh, one thing I'll just mention is that natural amino acids are mostly S, where we've got the NH2 in the carboxyl group poised this way, and this is usually a wedge with our side chain over here, where if we go 1, 2, 3, that's then S. Now we call these L amino acids because they have the same, what looks like configuration of L glyceraldehyde. It turns out it's not because they rotate light levorotatory. In fact, a lot of the amino acids actually rotate dextrorotatory, and so by definition, you probably should call them the D amino acids, but then that would confuse everything for the last 60 years of biochemistry research. So most of them are S. I like to draw them in this way, where nitrogen's pointed down, carboxy group is up and to the right on my screen, R group's up and to the left, and then we just have the nitrogen with the wedge. That's how I remember. You have to do some rotating as you flip things around. Hopefully you kind of caught that as I was rotating things around, I wanted to maintain the S configuration, which sometimes gave it a dash. Okay, anyway, that's long-winded enough. I'm going to stop talking for now, and we'll wrap up with this lecture. And according to my notes, this might be it for organic chemistry 2, at least for this year. So anyway, have a good one. It was a fun time. Enjoy watching the videos and being done watching the videos.